Hi plant propagators. This short unit is an introduction to some of the commonly used botanical terminology that's really useful to know before we start learning about vegetative propagation techniques. In this unit, we're going to look at some basic root and stem terminology and the concepts of totipotency and cell differentiation. And then we'll build on this knowledge in future units on the various vegetative propagation techniques that we're going to learn about. Those of you who've taken Hort 1A should already be familiar with most of the material in this unit, but it'll be a good refresher for you. By the end of this unit, you'll be able to describe the difference between taproots and fibrous root systems, identify the location of nodes, internodes, terminal and axillary buds on stems, explain briefly what a meristem is, define totipotency in plants, and explain what adventitious roots are. In the module on propagation from seed, we learned that the first structure to emerge from seeds is usually the radicle. The radicle is a small taproot. In some plants, this young taproot quickly develops secondary and tertiary roots, which then develop into a fibrous root system. In a fibrous root system, there are many roots of varying sizes, but no single root dominates in size, as you can see in the diagram on the far right. Examples of common plants that are around the Cabrillo campus that produce a fibrous root system include grasses, yarrow, mat rush, lomandra, and buckwheats. And here's a slide showing a couple of those plants. On the left, you've got yarrow, Achillea millefolium, and on the right is the commonly planted native grass, Muhlenberger rigens, or deergrass. In other plants, the initial taproot, that radicle, persists and increases in size, often growing really deeply into the soil. Smaller secondary and tertiary roots may develop from the taproot, but the taproot remains dominant in size, serving to anchor the plant in place, sometimes to tap into water sources deep in the soil profile, and sometimes acting as a store of carbohydrate. In plants with edible taproots, plant breeders have developed cultivated forms where the taproot is much larger than the naturally occurring wild forms, so the plants have become financially viable commercial food crops to provide food for humans. Examples include carrots, beetroot, parsnip, turnips, rutabaga, and sugar beet. The photo on the right here shows a young seedling of one of our locally native oak trees, coast live oak or Quercus agrifolia. In the photo, you can see the young taproot with its secondary roots at the bottom there that are developing. In the wild, the taproot of some of our native oaks can grow as deep as 70 feet in order to tap into water reserves that are held much deeper in the soil profile. The photo here shows a young pawpaw seedling with an air-pruned taproot and young secondary and a few tertiary roots that you can't really see very well. But you can see here that the taproot has a blunt end. Roots can't grow into dry air, so when this taproot grew out of the drainage hole at the bottom of the plug tray that the seed was planted in, the tip of the taproot died, and we call this air pruning. Nurseries often use air pruning techniques to prevent the development of a taproot and encourage early development of a more fibrous root system that will eventually produce a better quality nursery tree. The crown refers to the area at the base of the stem that forms the root-stem interface. It's usually right at soil level, so here and here, and is basically where the roots and stems are joined. When we're transplanting plants, It's really important that the crown stay at the soil or substrate surface and that we don't bury it or plant it too deeply. The plant can rot if the crown is buried. Confusingly, the term crown is also sometimes used to refer to the canopy of a tree, so be careful not to confuse the two uses of this word, although the meaning should usually be clear from the context. In the picture on the right here, you can see part of a stem from an apple tree. This section of stem has several nodes, here, here, and here, and some more. A node is the area of the stem where one or more leaves are attached. The leaf may be attached to the plant by a small stem called the petiole. At each node, in the angle of the stem and the petiole, there's an axillary bud, which depending on the species will grow to form a vegetative stem, which is a stem with leaves, or a stem with flowers. The axillary bud can vary in size depending on the plant species, but on most shrubs and trees it can be seen fairly easily. When we're selecting stems to use as stem cuttings, we look for stems that have healthy axillary buds that don't look dried up or as if they've been chewed by insects. The section of stem in between each node is called the internode. The internode can be quite long or it can be extremely short, as you'll see in the next slide. The flat, expanded part of the leaf is called the leaf blade or lamina, and each leaf has an upper leaf blade and a lower leaf blade. At the tip of the stem, there's usually a bud, which we call the apical or terminal bud. In this slide, you can see photos of 3 different plants, illustrating how the length of the internode can differ. The stem on the left is from the woody shrub lemon bottlebrush or Melaleuca citrinus, and the internodes here are round about 1 inch to 1.5 inches long, and you can clearly see the axillary buds here in the angle of the petiole and the stem. The photo in the middle is of the succulent purple bush aeonium, whose leaves are arranged in a really dense rosette. The internodes here are barely 1 millimeter long, and we can only see them if we separate the leaves. The plant on the right is the South African herbaceous perennial Pelargonium sidoides, or South African pelargonium, which also has very short internodes. One of our skills as propagators is knowing where to make the cuts on plants with all of their differently sized internodes in order to make stem cuttings. Meristems are zones where plant cells are actively dividing by mitosis. Primary meristems are associated with plant growth. An example of a primary meristem is an apical meristem, which can be found at both the tips of shoots and the tips of roots. Apical meristems give rise to the main body of the plant and are responsible for the extension or growth of roots and shoots. A microscope is needed to see the cells within meristems. In the picture on the far right, you can see the location of the apical meristem in roots. There's an area of rapidly dividing cells just behind the protective root cap. These rapidly dividing cells develop into the various tissues which make up the plant root, and they extend the length of the root as they develop. The picture on the near right is a longitudinal section through the tip of a stem and shows the typical location of an apical meristem on the shoot of a plant. New meristematic areas, which are areas of rapidly dividing cells, can also be induced by wounding a plant. Wound-induced meristems are groups of cells that were already differentiated and then de-differentiate into new meristematic zones, which results in the regeneration of new plant organs. This is an important process in vegetative propagation techniques, where we need stem cuttings, for example, to develop new root systems and leaf cuttings to develop both new shoots and new roots. But let's talk a little bit more about differentiation so that we understand this concept a little more clearly. Organisms, both animal and plant, are composed of different cell types. As new cells develop, they become dedicated to a particular physiological function and are said to be differentiated. For example, muscle, nerve, and epidermal skin tissue in humans are all differentiated cells with a specific purpose. In most animals, including humans, once cells are differentiated, they can't de-differentiate, meaning they can't stop being cells of one type of tissue and then change to another type. In other words, once you're a muscle cell, you're always a muscle cell. In contrast, in plants, the cells of some plant parts do have the ability to de-differentiate when they're given appropriate stimuli. Totipotency is the term given to differentiated cells that have this ability to initiate cell divisions, de-differentiate, then redifferentiate as new meristematic growing points of a different type of tissue. These cells are described as being totipotent. Totipotency means all power. So basically, these cells have all the power to become cells of a different type of tissue. The differentiation and redifferentiation of totipotent cells are under genetic control. The genes need a trigger or a signal from plant growth regulators in order to start the de-differentiation process. In propagation, this trigger is activated by the wounding process created when we snip a cutting from the mother plant. Totipotency forms the basis of our ability to propagate plants vegetatively. If existing plant tissues weren't able to de-differentiate, then form new meristems, then redifferentiate as new root tissue, vascular tissue, and stem tissue, then we wouldn't be able to propagate plants vegetatively. Let's talk now about roots again. The roots produced by vegetative propagation methods, such as cuttings, are referred to as adventitious roots. Basically, adventitious roots are roots that are produced anywhere on the plant other than from the original crown. Some plants have preformed or latent root primordia. Primordia is a term used to refer to a plant part or organ in its very earliest, almost undeveloped state of development. These root primordia are sometimes referred to as root initials. The preformed root initials develop naturally on stems while they're still attached to the parent plant. Plants that produce preformed root primordia usually produce adventitious roots easily and quickly. And common examples include willow, poplar, hydrangea, and jasmine. The roots of some plants with preformed root initials may even emerge without any wounding occurring, and some of you may have seen plants that do this. Common examples include quite a lot of penstemons, tomato, as you can see in the photo on the right here, and the succulent bush aeonium, Aeonium arboreum. Not all plants have preformed root initials, though. Many plants only form adventitious roots in response to wounding. In these cases, we say that the roots are formed de novo, which is Latin for from new, or basically the roots are formed from scratch. The ability of cuttings of all types, stem cuttings, root cuttings, leaf bud, and leaf cuttings, to form adventitious roots, new vascular tissue, and shoots is based on the concepts of totipotency and differentiation. So let's just summarize what we've covered in this short unit. In this unit, we've identified some of the basic plant vegetative structures which will help determine how to prepare cuttings and divisions. The ability of cuttings to develop new roots and shoots is based on some plant cells being totipotent. Totipotency refers to differentiated cells which have the ability to dedifferentiate, form new meristematic zones where the cells can redifferentiate as new plant tissues, such as adventitious roots. A wounding stimulus is often needed in order for this dedifferentiation process to occur. Adventitious roots are roots which are formed anywhere on a plant other than the original crown. Some plants have preformed root initials and produce adventitious roots very easily, sometimes without any wounding process being necessary. Other plants don't have preformed root initials and will only produce adventitious roots in response to a wounding process, which stimulates the production of plant growth regulators that then initiate the rooting process. So that's the end of this unit. When you have time, head over to the short quiz to test your understanding of the information covered in this unit.